Here's an interesting brain disorder. It's called Lissencephaly, which literally means smooth brain. And it's a rare brain formation disorder that's caused by problems in the development of the brain. It's thought that defective neuronal migration during early brain development is what causes this lack of folds and grooves in the brain. So you see that there are no sulci and gyri. Children who have Lissencephaly generally have significant developmental delays. Often they experience seizures or other physical problems. But these vary greatly from child to child, and some individuals with Lissencephaly appear to have no cognitive difficulties. The causes of Lissencephaly are not clear, but this has been linked to viral infections during pregnancy, and there's some evidence that it may actually be genetic as well. Cancerous tumors, or cells that grow uncontrollably in the body and serve no useful function, are another type of brain disorder. There are essentially two types of tumors, malignant and benign. And the main distinction between them is whether the tumor is encapsulated, whether there is a distinct border between the tumor and the surrounding cells. So benign tumors are encapsulated. There is a distinct border that surrounds the tumor and prevents it from spreading to any of the surrounding tissue or infiltrating the surrounding tissue. Benign tumors can be removed relatively easily with surgery and will not return. Malignant tumors, on the other hand, grow by infiltrating the surrounding tissue, and there's nothing preventing the malignant tumor from spreading to the tissue around it. It can be removed in surgery, but any cells left behind will regrow into another tumor. And also, these kinds of tumors are able to metastasize, so cells from the tumor will break off and travel to other parts of the body to grow. Malignant tumors do not have that encapsulated border, and that's why they're able to infiltrate and overtake surrounding tissues. That's what makes them malignant. Now, any tumor can cause brain damage by compression as it grows. Even a benign tumor displaces space, and that leaves the potential for damaging healthy neurons that it displaces as it grows. It can also damage those healthy neurons by blocking the flow of cerebrospinal fluid and producing fluid on the brain. Malignant tumors can also damage brain tissue by spreading and infiltrating, as I mentioned before, into the surrounding tissues. That infiltration into the surrounding tissues will kill the neurons and destroy that tissue on the way as it grows. Brain damage caused by a tumor is dependent on the area that's affected. So if the frontal lobes are affected by the brain tumor and there's damage in that area, then you would expect a loss of function that is associated with the frontal lobes, such as planning, attention, understanding the effects of your actions on others, for example. Here's an example of a brain tumor, specifically a meningioma, because this tumor originated in the meninges, that protective covering around the brain. And this particular tumor was causing vision problems, among other symptoms, because it was compressing healthy tissue in the occipital lobes. Brain tumors can also compress arteries, cutting off blood flow to other healthy brain tissue and causing neuron death there and loss of function. So damage that's caused by brain tumors is generally related to the structures and the healthy tissue that is affected by the tumor. Brain tumors can be treated by removing them with surgery or with radiation. Newer treatments for cancer and for tumors are focused on drugs that stop the development of new blood vessels, because it's the blood vessels that grow to support those new tumors. So it's thought that if drugs can block the blood vessels from growing, then it can starve the cancerous tumors. Next, I'll talk about degenerative and disease-related brain disorders. The first of these is what used to be called epilepsy, seizure disorders. Seizure disorders affect 2.5 million Americans. There's been a shift by physicians from calling this disorder epilepsy to calling it seizure disorder, because they've wanted to reduce the negative stigma associated with the term epilepsy. A seizure is a sudden, uncontrolled burst of electrical activity in the cerebral neurons. The brain is constantly discharging electrical activity in a controlled manner, and that's what keeps the brain functioning. But occasionally, there is uncontrolled firing of the neurons in the brain, and when the motor neurons are involved, the result can be convulsions. Sometimes right before a seizure, a person will experience an aura, and an aura is a sensation that precedes a seizure. And the symptoms of the aura depend on the origin of the seizure in the brain. So whatever part of the brain that is the focal point or the origin of the seizure, if you were to electrically stimulate that part of the brain, the person would experience the same sensation. Because a lot of tumors originate in the temporal lobe, often people experience a sensation that you would expect if you stimulated parts of the temporal lobe near the amygdala. They experience a feeling of fear or dread. Seizures can be the result of brain injury, infection, withdrawal from alcohol or drugs, or even a developmental abnormality like a malformed blood vessel, or sometimes they can have no apparent cause. Seizure disorder occurs when these seizures occur repeatedly. Seizures can be either partial or generalized, which means they can either be localized in one part of the brain and not spread, those are partial seizures, or they can spread to other areas, and that's the generalized seizure. Many originate in the temporal lobe, as I was mentioning, and so it's not unusual for someone with a seizure disorder to experience that aura of intense emotions right before the seizure. In 50% of those who have seizure disorder, there is hippocampal damage, and this damage becomes worse the longer the disorder and the more severe the seizures. I briefly mentioned some of the potential causes of seizure disorders, but another cause is withdrawal from depressants such as alcohol or barbiturates. That can cause the brain to be in a hyper-excitable state and produce seizures because the brain is so used to being inhibited by the drugs. So treatment for seizures has focused on drugs that increase the inhibitory firing in the brain and also surgery to remove the source of damaged tissue. Just recently, another type of treatment for seizure disorder was approved, and that is an implant that's put just underneath the scalp, and that implant is designed to fire as soon as it senses a seizure coming, and it sends an electrical signal to short-circuit the seizure before it even begins, and it's been shown to be very effective. Another kind of brain disorder, sometimes referred to as a stroke, is cerebrovascular accidents. And a cerebrovascular accident results in a rapid loss of brain function. This can be due to ischemia, which is lack of blood flow caused by blockage or by a hemorrhage. Ischemic strokes, those caused by blockage, usually are caused by blood clots that have formed in the blood vessels that block blood flow, or by other debris that is broken off and lodged in an artery that's too small to pass through. And as a result, it blocks the delivery of oxygen and glucose to the cells. Hemorrhagic strokes often result from high blood pressure and the effect of the high blood pressure weakening the blood vessels in the brain. Eventually, the blood vessels in the brain give way, and the blood that seeps out accumulates and puts pressure on the surrounding neurons, damaging them. As a result of the stroke, the affected area of the brain is not able to function. Now, this might result in the inability to move one or more limbs on one side of the body, or an inability to understand speech or to be able to speak, or a loss of vision. And that is dependent on where the stroke occurs in the brain. Damage from a stroke can be permanent, but the extent of the damage can vary with the size of the affected blood vessel. And treatment can be surgery to repair the damage, or it can be medication to reduce blood pressure and clotting. The number of people who are affected by cerebrovascular accidents in the United States tops 750,000. It's intuitive to think that neurons would die in a stroke because they're being cut off from blood, glucose, and oxygen. So essentially they die of starvation. But the actual cause of death is not that starvation, but it's excessive amounts of glutamate, which is excitotoxicity. Loss of oxygen and glucose results in a failure of the sodium and potassium transporters in neurons. And so what happens then is that the neurons can no longer maintain their resting potential. And the insides of the neurons become completely depolarized indefinitely. And this depolarization results in the release of glutamate, and the activation of glutamate receptors causes more sodium ions to enter the cell and causes the neurons to absorb excessive amounts of calcium through the NMDA channels that glutamate is opening. When a cell is full of excess calcium and sodium, it's toxic, and that's what kills the cells. The sodium causes the neurons to absorb water and swell, and other changes take place in that neuron that contribute to its death. So drugs that are directed at preventing glutamate from binding with the NMDA receptors, NMDA antagonists, have been shown to reduce damage caused by stroke, since the damage is caused by that excitotoxicity or that release of glutamate. These kinds of accidents can be prevented with lifestyle changes that can reduce plaque forming on the insides of vessels, and also by reducing blood pressure. Those two things can reduce the likelihood that a person will experience a cardiovascular accident or a stroke. Vascular dementia or multi-infarct dementia is another brain disorder that results in dementia due to problems in the supply of blood to the brain caused by a series of minor strokes. Minor strokes cause small lesions in the neurons in the brain because blockage of the blood results in cellular death. You can see evidence of dementia in a brain scan by noticing the lighter spots in the scan. Those indicate damage from small strokes. So you can see in this image, the arrow is pointing at the small, lighter areas in the brain, and those are places where damage has occurred from small strokes. This type of dementia was previously called multi-infarct dementia and also hardening of the arteries. Vascular dementia is the second most common dementia after Alzheimer's. When mad cow first made the news in the 1980s and 90s, it brought attention to another type of brain disorder. This one's caused by a contagious disease called transmissible spongiformencephalopathy. The outbreak of mad cow was actually caused by bovine spongiformencephalopathy. This disease results in a brain that has tiny little holes, so when you look at it on a slide, it looks like a sponge or Swiss cheese. Eventually, they learned that this disease was not caused by a virus or a bacteria, but by a simple protein called a prion, a protein that everyone has naturally in their bodies that had gone bad. So what are prions? Prion was named prion by its discoverer, a guy named Prusiner, because it was short for protein infectious agent, although I'm not sure how he got there with that name. But the prion is a protein that exists in the membrane of neurons normally. The only difference between a good prion and the ones that cause this degenerative disease is how they're folded. Misfolding of prions makes them have a different three-dimensional shape, and that three-dimensional shape is what actually does all the damage in the neuron. So prions are actually supposed to have the shape of the ones on the left, the normal prion protein, the curly structure. However, those proteins misfold, and they become straightened sheets of protein rather than curly, like the ones on the right. These bad proteins in the neuron produce transmissible spongiformencephalopathy because once a bad protein enters a neuron, it causes all the other proteins to become misfolded too. It's like a really bad influence. And it's this conversion process of converting all of the normal prions to this bad version, this abnormal version, that actually kills the neurons and causes the holes in the brain that look like Swiss cheese. There are other diseases before mad cow that actually were spread using prions, and the first of these was Scrapy. Scrapy was originally discovered in sheep and goats over 300 years ago. Sheep and goats became ill with the disease that caused such intense itching that they would rub themselves against something and end up scraping their fur off, hence the name Scrapy. However, this disease was not a threat to humans because humans could eat the meat of infected animals and they didn't become ill with Scrapy. Later, the first human TSC was identified by two physicians who named the disease after themselves, Creutzfeldt-Jakob disease. And symptoms of this disease include impairment in sensory functioning, cognitive processing, speech and movement, dementia, and eventually death. This disease is very rare, and its cause is thought to be both genetic, it was found to run in families, but also from a spontaneous genetic mutation in families with no history of the disease. Both forms of the disease are contagious, and they can be transmitted to others by transplantation of infected tissue. For example, one person unknowingly received an infected cornea from a donor and then later contracted Creutzfeldt-Jakob disease. It can also be transmitted by contaminated surgical instruments if they are unknowingly infected with prions, because current sterilization procedures are not effective against prions. Kuru is another TSC that was discovered in the 1950s among the Fore people of New Guinea. The symptoms of this disease were very similar to CJD, and eventually they were able to trace it back to a cannibalism ritual that the people engaged in when anyone died of the disease. When someone died of Kuru, out of respect, they had a ritual that included eating the brain tissue of the dead person. And eating that brain tissue allowed the prions to be transmitted from the person who died of the disease to each person that ate the brain tissue. The result was a huge number of the 4 people dying, especially women and children, who were the ones that most often participated in the ritual. When they realized what was happening, they abandoned the ritual, and the disease has almost disappeared. Here you can see the sponge-like or Swiss cheese-like look of actual slides of brain tissue that were infected with TSC. And in each of these pictures you can see those characteristic holes, that Swiss cheese or sponge-like appearance of brain tissue that has been infected with prions. So some have speculated, what is the purpose of the prions? One of the theories is that something like a prion could play a role in the establishment and maintenance of long-term memories. Long-term memories can last for decades, and prion proteins, which are resistant to the destructive effects of enzymes, may maintain those synaptic changes for long periods of time. So they may actually play a role in memory. Parkinson's disease, a degenerative brain disorder, involves progressive difficulty in voluntary movements, muscular rigidity, postural instability, and sometimes includes tremors when a person is resting. Now, these tremors diminish when the person tries to move, but as soon as they rest, they return. And people with Parkinson's disease also appear to have a stooped-over posture. This disease was named by a doctor by the name of Parkinson, and it is seen in approximately 1% of people over 65 years of age, more males than females. It's thought to be caused by degeneration of dopamine neurons in the substantia nigra in the brainstem. The substantia nigra is closely connected with the basal ganglia in the cerebral hemispheres, and they're extremely important in voluntary movement. When the neurons that are involved in that communication, the dopamine neurons, are destroyed, then communication between the brainstem and the part of the brain that's involved in movement is disrupted. And that's why individuals with Parkinson's have those tremors and difficulty with motor movement. The breakdown of dopaminergic neurons in the substantia nigra has a devastating effect on a person's ability to move or to stay still. Parkinson's is thought to have genetic links, especially when it occurs early in life, as is the case with Michael J. Fox. But other things have been shown to be related to Parkinson's disease as well. There was an unfortunate natural experiment when several young heroin addicts were exposed to synthetic heroin, and they suddenly developed symptoms of Parkinson's. The heroin contained a chemical that became toxic and suddenly degenerated all their dopaminergic neurons in their substantia nigra, and that's why it produced the symptoms like Parkinson's. Other environmental toxins have been shown to produce similar symptoms. For example, insecticides and herbicides that are commonly used in gardening have been shown to produce tremors like Parkinson's. And also, if you have a head trauma that results in a loss of consciousness, you're 11 times more likely to develop Parkinson's disease later in life. So there seems to be a relation between head trauma and Parkinson's disease. Happily, drinking caffeinated coffee seems to reduce the risk of getting the disease, but they're not really sure why that is. Parkinson's is treated with drugs that are designed to increase the amount of dopamine in the brainstem. So L-Dopa, which is the precursor to dopamine, is given to increase the amount of dopamine that can be produced in the remaining neurons in the substantia nigra. And this usually works well at first, but eventually it loses its effectiveness over time. And it also has an unfortunate side effect. Producing more dopamine in the substantia nigra is good for people who have Parkinson's disease, but producing more dopamine in other parts of the brain, such as the mesolimbic mesocortical system, has the effect of producing psychotic side effects, such as hallucinations and delusions. So drug treatment has its limitations. Other treatments have included transplants of fetal cells and stem cells, designed to increase the number of dopaminergic neurons in the brainstem, gene therapy, surgery, and also deep brain stimulation that involves stimulating parts of the thalamus to help in that communication and to reduce the movement disorders that results from the lack of signaling from dopamine. So degeneration of dopaminergic neurons in the brainstem, in the substantia nigra of the brainstem, reduces the ability of an individual to initiate voluntary motor movement. So the degeneration of dopaminergic neurons results in a shortage of dopamine that impacts that person's ability to be able to initiate voluntary motor movement because they're less able to signal from the brainstem to the basal ganglia to initiate that movement. So this diagram shows a dopaminergic synapse, and the effects of having Parkinson's disease are shown in the bottom half of the picture. There's a great reduction of dopamine in the synapse. and that reduction of neurotransmitter reduces the amount of communication that can occur between those neurons and other parts of the brain that are involved in movement. So therapy is designed to increase the amount of dopamine in that synapse. In the picture on the left, you'll see an example of a Lewy body. Lewy bodies have been found in those dopaminergic neurons that remain in the substantia nigra. And Lewy bodies are structures that have a buildup of protein in them. Unfortunately, as of now, it's currently not known what triggers the destruction of dopamine neurons in the substantia nigra that causes Parkinson's, and it's also not known what triggers the creation of these Lewy bodies in the neurons that are left in the substantia nigra. Huntington's disease is another disease that involves the basal ganglia. Huntington's disease is an inherited disorder in which the caudate nucleus and the putamen of the basal ganglia degenerate. This inherited disorder is a result of a dominant gene, so all you need is one gene to develop this disease. And that one dominant gene produces a defect in the encoding of Huntington protein, which is a protein that is common for all humans. And the defect in the encoding of that Huntington is what causes the breakdown of the basal ganglia. Whereas Parkinson's disease causes a poverty of movements, Huntington's disease causes uncontrollable ones, especially jerky limb movements that look like they're purposeful, but they're fragmented, and they occur involuntarily. The first signs of neural degeneration occur in the putamen in a group of GABA neurons, and the result is that it reduces the ability of those neurons to inhibit the motor areas of the frontal cortex, and it results in those involuntary movements. This disease used to be called Huntington's chorea because those involuntary movements are also called chorea. Degeneration also occurs in other areas of the brain. It's not limited to the basal ganglia, including the cerebral cortex, and it impacts the person's emotions and personality as well. Eventually, it causes dementia and death. As of now, there's no treatment for Huntington's disease. So the most common form of dementia, which occurs in approximately 10% of the population for people who are above the age of 65 and 50% of the population for those who are older than 85, is Alzheimer's disease. Alzheimer's disease appears to be somewhat hereditary, but it is not clear what the cause of this disorder is. The symptoms include progressive memory loss, motor movement problems, dementia, personality changes, and eventually death. The initial signs might be difficulty remembering appointments or having trouble finding words or having trouble remembering other people's names. But as time passes, the person with Alzheimer's disease shows increasing confusion and more difficulty with simple tasks, simple everyday tasks like balancing a checkbook. The memory deficits are more severe in Alzheimer's for more recent events, so it appears to spare long-term memory more than short-term memory. Alzheimer's disease results in degeneration and deterioration of the hippocampus and other areas in the brain. And the brain of a patient with Alzheimer's disease, if you were to look at an image of it, has much wider sulci, especially in the frontal and temporal lobes, and the sulci are the grooves. And so if the grooves are much wider, then that means that there's substantial loss of cortical tissue. One of the hallmarks of Alzheimer's disease is that if you look at their brains following death, and this was the only way you could actually definitively diagnose Alzheimer's disease, you would be able to see the characteristic plaques and tangles, which are the neurofibrillary tangles and the beta amyloid plaques that are present in neurons when an individual has Alzheimer's disease. So beta amyloid plaque is a form of plaque that's found in the brain, and it's a plaque that everyone produces, but it's usually carried out of the body on a regular basis for younger, healthier people. In Alzheimer's, though, the brain produces excessive amounts of this beta amyloid plaque, and that plaque consists of really dense proteins. And those dense proteins eventually start interfering with cellular communication, and that results in the damage and the death of neurons, because if neurons aren't able to communicate, they eventually die. And when those neurons die, normal processes in the brain eliminate the leftover neuron, but a core of that beta amyloid plaque remains in the brain. Now, while beta amyloid plaque creates problems outside of neurons, a protein called tau can corrupt neurons from the inside. So tau protein normally exists in neurons, and it enhances the transmission of signals along the axons and the dendrites. It's kind of like a track, and it runs through brain cells. It's also been described as the neuron's skeleton. If a tau protein becomes toxic or misshapen, the tracks collapse and they form these neurofibrillary tangles. These tangles cause the communication between cells to break down, just like the beta amyloid plaque does, and that also leads to the eventual death of the neurons. Now, research with people with Alzheimer's has shown that these neurofibrillary tangles first occur near the hippocampus, and it's thought that Alzheimer's originates with atrophy of the hippocampus, which then spreads to the frontal, the temporal, and the parietal lobes. So for people with Alzheimer's, these plaques and tangles begin accumulating long before the symptoms of Alzheimer's are present. But it wasn't possible to identify those plaques and tangles until an autopsy, because then you could actually visualize those plaques and tangles in the neural tissue. But recently, researchers have developed a brand new method that is able to spot beta amyloid plaques. They've developed a dye that can be injected, and it goes into the brain and actually attaches to the beta amyloid plaques. After the dye has been injected, you can do a PET scan, and the dye that is attached to beta amyloid plaque, if it's there, will light up in the scan and reveal that amyloid plaque. Here's an example of this new method in which they've injected a dye that sticks to amyloid plaque and then done a brain scan using a PET scanner, and the PET scan picks up the dye and will show you exactly how much and where the amyloid plaque is in the brain. So on the left you see a normal brain, and on the right you can see the clear difference that shows the presence of the amyloid plaque in the person's brain. The last brain disorder I'll talk about is multiple sclerosis, and it is an autoimmune demyelinating brain disorder. So what happens with this disorder is that at locations in the central nervous system, a person's own immune system starts to attack their myelin. And myelin, as you know, is that sheath, that fatty layer that insulates axons and allows axons to transmit neural impulses much faster. When the immune system attacks them, it leaves hard patches or scarred debris called sclerotic plaque, and it's actually scarring of the myelin. It has the effect of reducing the ability of the nerves to communicate. This destruction of myelin causes an interruption in that normal transmission of neural messages through the axons, and when it gets to the ones that have been demyelinated by the disease, the communication is impaired. In multiple sclerosis, the destruction of myelin occurs in scattered places in the brain and the spinal cord, and so it produces a multiple number of neurological disorders. No two people experience MS in the same way, and often symptoms will come and go. The disease is more common in women than men, and usually occurs when a person is in his or her late 20s or 30s. So symptoms include blurred vision, visual problems, difficulty concentrating, numbness, weakness, and tingling in the limbs, difficulty walking, cognitive problems, dizziness, and fatigue, as well as difficulty with speech. But no two people experience MS the same way, and symptoms often come and go. As of now, the cause of MS is still unknown. And that's the end of this lecture.